We've talked about the organization of muscles, the muscle, the fascicles, the muscle fibers, the myofibrils. We spent time looking at all the different parts of the myofibrils, all the bands, the A bands, the I bands, the M lines, the H zones, and all the Z discs and all that stuff. It's time now to start looking up close at the thick and the thin filaments themselves, because that's where contraction actually happens. So you can look and see here, those structures made of blue balls. I mean, here I've been teaching for 45 years and I have to talk about blue balls. But the thin filament is made up of individual molecules of a protein called actin. So that's what you're seeing. The thin filament, each one of those little blue balls, is a molecule of actin, a protein. Then the thick filament is made up of molecules of myosin. And myosin is an interesting protein. They have heads that look like golf clubs. So that's what you're seeing all over those thick filaments. All those golf club-like heads are on the ends of the myosin proteins. Again, so thin filaments made up of actin, thick filaments made up of myosin. So how do muscles actually contract? How do these things cause contracting to take place? Well, see, those golf club-like heads on the myosin are actually going to grab the thin filament. And what's going to happen next? See if you can figure it out. Yeah. So contracting a muscle is kind of like rowing a boat. That myosin head is going to grab the actin filament, and it's going to do a rowing motion, and that's going to pull the thin filament past the thick filament. That's what's actually going to take place, all right? There you see lots of pictures of people rowing boats. That bottom picture in the middle, you know, that's a very famous boat. You know what that is? Yeah, that's one of the lifeboats from the Titanic. You can see Kate Hudson in the boat, and there's Leo DiCaprio's dead body out there in the water. It's not easy to see. If you look hard, I think you can see it. So what happens in skeletal muscle contraction? Here's a little sort of animation if you do the animation inside of your head. If we go from upper left to lower right, it's what we've seen before, but just again a chance to see it. The thin filaments are pulled towards the M line, okay? So that's what's happening. The myosin heads will grab those thin filaments. The H zone will get smaller. See, notice in the upper left, the H zone is, see how wide it is? And then by the time we get to the middle, the H zone is narrower. By the time we get to the bottom right, look, it completely disappears. There is no H zone anymore. And then look at the Z discs in the upper left. See how far apart they are? In the middle, they're closer together. Look in the bottom right. Now they're much closer together. So that's what happens. And the I band shrinks. The amount of the light-colored band gets smaller and smaller, all right, as the thin filaments get pulled past the thick filaments. Okay, once again, just a cartoon, again showing the same thing. That's what actually happens. Notice the Z lines, how far apart they are at the top and how close they are at the bottom. That's what happens. Each sarcomere gets narrower, all right? Remember, sarcomere goes from Z line to Z line. And as each sarcomere gets narrower, the entire muscle does, okay? Let's look at the individual muscle proteins. Myofibril is made up of three main kinds of proteins here. Contractile proteins, which actually do the contracting. They generate the contractile force. Regulatory proteins that switch it on and off, like a light switch. Again, remember, muscles are not contracting all the time. All the muscles in your body are not constantly contracting. They only contract when something tells them to, and so it's a regulatory protein that's going to tell them to contract. And then the structural proteins are basically just the 2x4s and the concrete. It just holds everything in place, okay? So, again, some proteins do the actual contraction, other proteins regulate when that happens, and other proteins simply make the whole structure. The two contractile proteins are myosin, which make up the thick filaments, and actin, which make up the thin filaments. All right, so we've been talking about myosin and actin. There they are. Those are the contractile proteins. Regulatory proteins are troponin and tropomyosin, and we're going to start talking a lot about those coming up. Troponin and tropomyosin are the regulatory proteins. Those turn contraction on and off. The structural proteins we've mentioned before a little bit. Titin, I think I probably want you to know that that's what stabilizes the Z discs. Myomecin is what runs down the middle of the sarcomere. That kind of holds the whole thing together. Kind of like you had a big bundle of twigs and you tied them together with a piece of string. Nebulin, I don't care about so much. Dystrophin is interesting because dystrophin helps hold the sarcomere together. Some people are born with a genetic defect and they don't have properly formed dystrophin proteins, and you may know what that disorder is called. Right, muscular dystrophy. In fact, there are a bunch of muscular dystrophies. The most common one is Duchenne's dystrophy. So zooming in here on muscle proteins. F-actin is the filamentous actin, and G-actin is the globular actin. So remember back to the proteins, the filamentous ones were the structure, the globular were often the function, they actually were the workers. I'm not going to make a big deal about this from this point on. The G-actin has the active binding sites for the myosin heads. We're just mostly going to talk about actin. I promise that I will not ask you to distinguish F-actin and G-actin from this point on. But I just did want to show you that in fact they're slightly different. They're two different. They merge together, and we're just going to call them actin. Okay. Nebulin holds the F-actin strands together. I won't ask you that again. I promise. You don't have to know that. Tropomyosin here we do have to know. Tropomyosin and troponin. So tropomyosin is the filament. So look in the bottom left. You can see the filament, the strand of tropomyosin. And then troponin are those blue blobby things. Those are proteins as well. Troponin is a globular protein that binds to the tropomyosin. Now, the fact is that although they're separate, they always function together. Troponin and tropomyosin always move together. So what I'm going to start doing is I'm going to be calling it the troponin-tropomyosin complex. And I'm promising that I won't ask you specific questions to make you distinguish troponin from tropomyosin. But you do have to know what the troponin-tropomyosin complex does. You have to know what both of them do together. And it's very easy. So it's all going to be regulated by calcium. I've talked before about how important calcium is in muscles, and it certainly is. Muscles don't contract without calcium. So what we're going to see here is that calcium is going to bind to troponin and cause a conformational change in the troponin-tropomyosin complex, exposing the myosin binding sites on the G-actin. Now that was a mouthful, wasn't it? Let's take a look at this next picture, and I think maybe it'll help. So look in the upper left. You see there the myosin head, all right? That kind of flesh-colored thing. And there you see we're back to the blue balls here for actin. And then you see the strand of troponin, the kind of, again, flesh-colored thing. And the tropomyosin, the troponin is the yellow. So notice how the myosin head is trying to bind to the actin molecules, all right? The big flesh-colored golf club is trying to get to the blue balls. But the troponin-tropomyosin blocks it. It's in the way. It can't get there. And that's the way muscles are when they're at rest. The troponin-tropomyosin complex keeps the actin head, the myosin head, from grabbing the actin. Now, let's look in the lower left, okay? So you see the blue actin, you see the troponin, tropomyosin together, all right? See the yellow and the little flesh-colored thing, that's the troponin-tropomyosin. And there you can see the myosin head up on the top. So again, on the far left, the very far left, you can see the myosin head wants to get to, on the blue actin molecule, there's that little black dot, that's the myosin binding site. The myosin head is trying to get there. But look, the troponin-tropomyosin is in the way. It's blocking access. So here's what happens. So then look at part B of that illustration. Here comes a molecule of calcium. And what does it do? It causes the entire troponin-tropomyosin complex to rotate out of the way. So that's what we're seeing in C. That troponin-tropomyosin complex has now rotated out of the way, and now that myosin head can actually get to that black dot on the blue ball, which is the myosin binding site on the actin. And that's what we see in D. The myosin head has finally grabbed a hold of the actin molecule. So that's what's going on. That's why calcium is such a big deal. Normally, when a muscle is at rest, The myosin head cannot get to the actin, therefore there can be no contraction. It's only when calcium shows up that it pushes, causes the troponin tropomyosin to rotate out of the way, and now, by golly, that myosin head can grab that actin filament. And that's what we're seeing in this diagram. You can see it's two strands of myosin that bind together. They twist together, and then you got the two myosin heads there. And again, you can see up on the top the blue actin molecules and the troponin tropomyosin. It's blocking the myosin binding site. It's only when calcium comes into the act that we can actually get muscle contraction. So here I've tried to tell you throughout the class whenever you had to know numbers and when you didn't have to know them. Here's numbers you don't have to know. But sometimes people are just curious. How many of these things are there? We've been talking about all these different things. How many of these? Well, there's about 2,000 myofibrils in every muscle fiber. Remember we said myofibrils made up of about 80% of the muscle fiber? So every muscle fiber, every muscle cell has around 2,000 myofibrils. Again, those are the contractile organelles. Those are what actually do the contraction. There are about 10,000 sarcomeres per muscle fiber. So remember, the sarcomere goes from Z disc to Z disc. The thick and the thin filaments don't run the entire length of the muscle. Instead, there's like 10,000 sarcomeres, end to end to end to end to end, that make up the length of an entire muscle. All right? There are 300 to 400 molecules of the actin and about 40 to 60 troponin tropomyosin molecules for every thin filament, and about 500 molecules of myosin. So again, you don't have to know these, just for the sake of curiosity. Sometimes people, would like a sense of scale. How many are these things? Well, that's how many they are.